Good afternoon, everyone. I welcome you all to today's session. The topic for today's session is natural products-based drug discovery, which will be taken by Dr. Sandeepan Haldar, sir. Dr. Sandeepan Haldar has completed his doctoral program from the Department of Chemistry, Indian Institute of Technology, Kanpur. In his first doctoral research in the Bar Ilan University, Israel, he has worked on field of physical organic chemistry under the supervision of Professor Shamayu Hose. After that, he has been awarded prestigious Indo-USA Fulbright Nehru Postdoctoral Fellowship from United States India Educational Foundation. To pursue postdoctoral research, the Department of Chemistry and Biochemistry at the Ohio State University, Columbus, under the supervision of Professor T.V. Rajan Babu. In this duration, he was also involved in the research field of asymmetric catalysis for the hydrovinylation reactions. He joined the Department of Visvesvaraya National Institute of Technology, Nagpur, in the year 2016 as an associate professor. At present, Dr. Haldar's research group is working in the field of organocatalysis, precisely hydrogen bond donor catalysis. The catalytic methods have been further utilized for the stereoselective synthesis of active pharmaceutical ingredients for different medicinal applications. I now would like to invite Professor Dr. Haldar, sir, to please come on stage and share your valuable insights with us. Let's welcome him with a huge round of applause.
Thank you, Ria, for a very nice and lengthy introduction. Again, good afternoon to all of you. As you can see, the title of my presentation is Natural Product-Based Drug Discovery. Now, based on these discussions, what is going on from tomorrow yesterday onwards, how is it going back? It cannot go back. No. It was started from the fragment-based drug discovery, structure-based drug discovery, medicinal chemistry, but I'll keep you in an error in a particular period where the medicinal chemistry was chemistry only. Okay? So that particular time period, there was no separate discussions of these drug discovery things. Chemist was working on different kind of natural products to get their structures, to isolate them, and to characterize them. So in order to have this particular objective in our mind, the content of the presentation will be like that: introductions, natural products, different classes and the functions, and their timeline of the drug discovery, their contributions for that, those sort of natural products. Then the extractions, isolations of the natural products, their characterizations, which is very much important. Then modern techniques which are actually being used in order to isolate the products, to know their structures, to purify them in a better way, and today's scenario of the significance of those natural products in modern drug discovery. So this way I'll be outlined, and as I said, if you have any kind of doubts on it, you can stop me, you can interfere in between, I don't mind. So once I say natural products, that basically coming from the nature. Now, what do we mean about the nature? Products can come from some plant sources, which is mostly abundant, that herbal source, any kind of plant-based natural products. But then again, another things will be coming, which part of that plant? Is it a leaves or roots or some bark, anything, right? Followed by, it might not be only restricted to the plants based, it can come from the microbes. Different kind of microbes can produce different sort of natural products. It can also come from different animals as well. So we need to keep in these aspects in our mind, that if you want to start the understanding the role of natural products, where from we should start. Okay, so this was the scenario, I would say, almost 100 years back. Now it might be of different things, mostly these natural products are termed as secondary metabolites, and it's a small molecules. Then once I am saying secondary, then you need to know what is the primary metabolites then. So these are all the sources, microbial worlds, animal sources, marine worlds, even from venom, some toxins can also give different sort of natural products. Let it be like some cytotoxic effect, but that also we need to learn in order to get their structure activity relationships. So this is overall scenario. What are the sources? It might be some eukaryotic things, prokaryotic, the fungi, that different kind of animals and all. To start with, as you can see, the lipids, proteins, carbohydrates, amino acid, vitamins, they are all naturally occurring. And these are the ingredients which is actually making the living cells. Without that, no living organism is possible. So they are actually the primary metabolites which is actually keeping to make our living constituents. Okay. Now once these primary metabolites are converting to some products, that is basically the secondary metabolites. So once these kind of things which are actually the originating the lives are converting to some another kind of products, but in a natural way, in a biosynthetic ways, not a normal synthetic ways, then the secondary metabolites are actually coming into the picture. So we need to understand how that is going on. So as you can see, like this sort of class of secondary metabolites, you can start from non-protein amino acids, some kind of organic acids. It might be terpenoids. These are the classes based on their structures. But today's world, we know their structures, but earlier we didn't have these sort of classifications. We only get the natural products from the plant or different kind of sources. Now it can have the quinones kind of structures, different alkaloids, amines. It can have the phenolic compounds, cyanogenic glycosides. They are the carbohydrate-based structures. You can also see the benzoic acid, which is very common. That can also be having different isomers or different derivatives. Those are actually naturally occurring. Even the cinnamic acid also can have different substituents on the aromatic ring. They are actually taking part and existing in nature to have a diverse sort of, the biological activities and all. So they are coming from some primary metabolites, and after the biochemical pathway or biosynthetic pathway, they are giving the secondary metabolites which are actually termed as natural products. Now this is the overall scenarios. It can be termed like based on their functionality. You can see the phenolic acids depending upon their functional groups. Phenols, and then there is an acid, there is a phenolic acid groups. It could be a flavonoids kind of derivatives. It might be a stilbenes, coumarin derivatives, lignans, tannins as well. Like some kind of cases, they are very small molecules, or it might be a macromolecules as well. You can see the tannins and others. Depending upon their different diverse, the structural features, their role and their synthetic part will also be different. So now to get the actual classifications or to get the segregations of those entire natural products, we can have them in order to get their structural core. You can see the alkaloids, which are all nitrogen containing compounds. So where the compounds are having nitrogen, it might not be good as well. Always they are not good. They might be toxic also. So we have classified it into that particular levels, that this kind of nitrogen containing compounds keeping an aromatic ring or a aliphatic long chain like the structural features will be the alkaloids. If you move on, they are the terpenoids. They are without nitrogen containing groups. Like the structure doesn't contain nitrogen, but maybe oxygen. They are very small molecules. They'll be containing, mostly they are the hydrocarbons, they are the olefins. They'll be having some functionality like that unsaturations, but without nitrogens. They are the terpenoids. There are the other secondary metabolites which are coming from the different animals. I can see there are polyketoids, like ketide group. They are the macromolecules attached with maybe some sort of carbohydrate residues. They are the polyketoids coming from the nature. They can also have the cyclamic acid, cyclamate type. Cyclamate acid is a biosynthetic pathway. They are the cyclamic acid is a like the sort of the secondary metabolite which is having a defined biosynthetic pathway which can also be having the proper functionality. So now depending upon their functional groups, depending upon to their different biological activities, we can divide or we can classify them in some ways. Then how that biosynthetic is taking place? Because if you consider nature, the biochemical procedure is going on, which is starting from the photosynthesis, then it is nothing but carbon dioxide, water, they are interacting with some sort of, the molecules like chlorophylls is present over there, some metalloproteins are there. So they are producing what? Glucose. Now that particular glucose carbohydrate is further transforming, that is going for glycolysis, they are going for the malate, chorismate, then ending on the amino acid tryptophans. So this way the biosynthetic process, and it is automatically going on, there is no like synthetic chemist or maybe that is very difficult to mimic as well. Like you can understand once nature is the big chemist, like once the things are going on, there will be some sort of conversions. Nature will create their own enzyme in order to get that particular process done. Okay, so these are the pathways by which that very small molecules like glucose can be converted to some variety of different like the secondary metabolites, or maybe it can convert to the other pathway with that glycolysis, then phosphophenol pyruvate, then again in the like in the cell mitochondria, those sort of biochemical process will be going on to have the acetyl coenzyme A, then the mevalonic acid pathway or the malonic acid pathway. These are all like some sort of understanding of their mechanism, like how the biosynthesis of those things are happening, right? Because the process, we are not having any kind of synthetic tool in the nature that will put the reactions maybe in 60 degrees centigrade that put some definite reagents. No, nature will do the things on its own way. So we are having the photosynthesis, we are having different kinds of process like the either bond breaking or bond forming. Glycolysis is a bond breaking process, so it will be moving on that particular directions and to maintain that biochemical sequences to give you the corresponding products. So the idea will be getting these sort of products are existing in the nature. So you are basically isolating them and figuring out what might be the possible mechanisms in order to get that particular product through those biochemical processes. So you are defining the biosynthetic route. They are not the synthetic route, they are biosynthetic route to get those primary metabolites, amino acids kind of things, maybe different kind of carbohydrates. Everything is forming in the nature, not even any kind of synthetic things are involved here. So these data informations, we can have a huge number of classified natural products. As you can see, the terpenes, there are 29,000 of terpenes which are there. But the good part is that, or the beauty is that they are all come under the single domain, that is the terpenes, having some very specific kind of property. They are having a molecular weight range, they are having some particular type of functionality. They comes under the these things of the terpenes. It might be derived that C5 precursor, that isopentenyl diphosphate, 12,000 alkaloids, they are derived from different sort of amino acids. Some cases they are the naturally occurring, some cases maybe they are the unnatural amino acids, but actually they are the nitrogen containing products, they are the alkaloids. It might be 8,000 of different phenolic compounds they are known. So all of them having phenolic OH groups and along with some other functionalities which are there and used as a secondary metabolites. Followed by there are non-secondary metabolite nitrogen containing, again non-protein amino acids, those amino acids which are actually not taking part to get a protein, to make a protein. They are the non-protein amino acids. They are having the cyanogenic glycosides or the glucosinolates. They are the terminology or the classifications of the entire variety of the natural products. Now if you move on towards the without nitrogen containing, that is the terpenoids and the phenolics, it might be depending upon the range or the molecular weight of those compounds. It might be monoterpene, diterpene, triterpene, sesquiterpenes, phenolics. There are having flavonoids, polyacetylens, polyketyls, phenyl propanoids. So once you are having the entire range of products which are automatically forming, they are all making by the nature in your hand. You can know their corresponding functionality, you can classify them based on their properties as well. Along with that, you can have which kind of elements are present over there, and you can also get their corresponding, like, the expected property or their biologies. So this way the natural products and immense significantly importance in order to figure out the biology of those kind of processes. So now coming to the terpenoids, or it is isoprenoid, like you can see the isoprene structure. It's a conjugated, you can say, a double bond. There's a 3 carbon involved. It's a hydrocarbon. Okay, that is the very beginning of that terpene class of natural products, that is isoprene, or it might be limonene as well. That is also a natural products. So you can start with the terpenoids. The name it's come from the first member of the class. It's isolated from the turpentine. You know that oil, turpentine oil, it's available in the market. So you are just getting the things, you are isolating. Now that is the origin. Once you know that these sort of products you can isolate from nature, and then you can characterize what kind of structure it is. And you can classify them in a particular type of domain to know their properties. So afterwards you can move on isoprenoid, since that isoprene basic unit is C5 building blocks. So it is not only a small molecules like 3 carbons, 5 carbons maybe, it is becoming a more number of carbons into that particular fashions. After the isolation, it was long back, you can see the 1930s. So the Nobel Prize chemistry, it was given, the common compound is an isoprenoid if it is derived biologically form and with or without the rearrangement. So how that particular are forming in the nature, accordingly you can isolate them, you can characterize them, you can propose the mechanisms, how that biochemical processes are going on in order to get that particular natural products automatically forming in the nature. Okay. So that's what the importance was there, but as you can see, it is long back, almost 100 years back. The type of chemistry was discovered or the Nobel Prize awarded because of those kind of significant research. But that time you need to understand the characterization facilities and other sophisticated instrument was almost nil. So you have to do what are the things you are having, you have to isolate the products, and not only the isolation, that has to be very pure. So purification is another aspect. If you isolate a mixture product, then you cannot define their properties or you cannot get the structural understanding of those small molecules. So accordingly, you can devise your techniques. Without that, you cannot move on. So this is the actually biology of different terpenes. Biological role, property, it might be volatile if the molecular weight is small. If it is 3, 4 or something, if it is more than 5 or so, it may not be volatile. It might be non-volatile as well. The beauty is that they are all flavors, they are fragrance, they are scent, all sort of perfumes, they contain the terpenes kind of products. It can have antibiotic properties. They can be there in different sort of hormones. It can membrane lipid, insect attractant, like some sort of things, pheromones kind of things. They actually attract the insects. They are having those sort of properties. And also they are antiphidans to the insects. So mediate the electron transport process can also be possible with that. But all the cases, the structural similarities are there. They are not nitrogen containing, they are only oxygen containing. So depending upon their size, depending upon their number of carbons, it can be volatile or non-volatile or maybe deciding their corresponding boiling points. So this way it goes. Now some of the cases, these terpenes can be isolated under the ground from the roots of the plants, or it might be on the leaves of the plant. So accordingly their property might be changing, okay? But the class are same. Every cases they are the terpenoids. So this is the basic understanding of that. Now to start with, this is the hemiterpenes, it's a prenol. Now look at it, it's very simple structures. It's a prenol and they are actually coming from the potential sources of the biofuels. What are the things you can see, the natural occurring products which are actually having the property to get it for the fuel use, they are this sort of terpenes. It can have angelic acid or isovaleric acid or tiglic acid. Every cases the number of carbon is only 3 or 4 maximums. Functional groups, no nitrogen containing things are present, either hydroxide or carboxylic acid, and maybe one unsaturation is there. Depending upon their number of carbons, that is termed as hemiterpenes. The similar role or similar significance, they might have the biofuel property, or they might be used as a biofuel. Now followed by the monoterpenes, as you can see, they are the citrals, myrcene, ocimene, these sort of things, and here also the number of carbon is increasing, but there are no nitrogen, mostly the hydrocarbon. Or it might be having some terminal functionality, like in the first case citral, you see there is an aldehyde group present. Accordingly, you can define their corresponding properties. So they are acyclic in nature, there is no cyclic structures involved. Second things you go, carbon, they are the monocyclic monoterpenes, okay? They are the carbones, these sort of structures. Now the second one, that is menthol. See, yesterday also we had a discussion, there is a presence of the chiral center. Now because of that chiral center, the property is varying, and this sort of menthol, they are no more volatile, they are solid compounds. Followed by the D-limonene, that the stereochemistry is fixed. Now these particular chiral centers are having a particular stereochemistry, and that is built by the natural process. Okay, so these are the monocyclic monoterpenes. It might be bicyclic monoterpenes like alpha pinene, alpha thuzone, beta thuzone kind of things. They are every cases non-nitrogenous compound with some oxygen functionalities, or maybe the olefinic groups. It includes, as I mostly the components are the essential oils, plants, aroma, flavor. Is a vital range of ingredients for different kind of applications like agricultural, pharmaceutical, cosmetics, food applications. Okay, so these are the class of compounds which are monoterpenes. They are having a wide range of applications, but you have to choose that particular compound in order to achieve that particular type of applications. Now moving to the diterpene, you can see that automatically the number of carbon is getting increased in the structures. It might be having a long chain aliphatic groups, or it might be having again the chiral center may or may not present, but some of the cases they are the bicyclic or even tetracyclic rings are present. Okay, so you can see the molylactone A, that is a tetracyclic diterpenes, the non-volatile, almost C20 hydrocarbons derived, 4 isoprene units and structurally diverse. So they are the essential for the biological activities with agricultural secondary metabolites with potential production of the biopesticides. They are actually may give the biopesticides from these kind of compounds. Now followed by the triterpene, again the first one is squalene. It is simple and hydrocarbon, and it is there, it is naturally occurring. Bicyclic, that alpha poly podateterine, then tetracyclic, or it might be pentacyclic as well. These sort of things are having C30 precursor squalene. Over 20,000 of these kind of compounds are known today. Okay, so they are been isolated from the nature, they are been properly purified, they are been characterized, they are been classified in a particular way in terms of knowing their different sort of chemical as well as physico-chemical properties. They are different biological activities, they are structural activity relationships, those things are well established. Now to move forward towards the sesquiterpenes, furnish you all that this sort of alpha bisabolol, that note ketone, and these kind of things. You can see there is that 3-membered ring is also involved in the bridging positions. So accordingly the structural things are changing. Mostly there are 15 carbon skeletons. That's what they are no more volatiles. They are having huge molecular weights, and most of the cases they are containing the cyclic rings. Tetraterpenes, again big, you know the beta carotene presence in carrot. These are structures, they can be isolated, and along with their flavors they might have the color as well. Because of their structural features, you can see that highly conjugated structures. Depending upon that, their absorption things will be changing. They can absorb light in a particular wavelength, and again the emissions things will be different. So based on their structures, they are all basically either almost hydrocarbons, that beta carotene, lycopene, there are no heteroatom present over there. But very ordered and very proper way the structural things are there in order to have that particular type of activities. They are mostly C40 the carbon are present, and the carotenoids class of compounds are there. Okay. Now, as I said, they have been classified, they have been synthesized in nature, so we need to know how that biosynthetic things might be possible, because you cannot actually like figure out the mechanisms, but depending upon the structures of the products and which particular source it might be forming, you can just get that what might be their corresponding mechanism of the synthesis. It's generating from the phenyltransferases. These are the enzymes that nature is automatically developing for do that particular process done. It might be the GPP or FPP or GGPP, these sort of enzymes. Terpene synthase reactions. Synthase, that is the enzymes which is actually operating on it. Monoterpene synthase or cyclase, and the modification is one enzyme, one substrate, multiple products. So once you are having a substrate, you put the enzymes, but you can get a wide variety of products through that biosynthetic pathway. So cytosol kind of mechanisms, FDP synthase, or it might be a plastid kind of mechanisms, the GDP synthase, which will be ultimately giving you the secondary metabolite, this farnesyl diphosphate or general diphosphate kind of products from there. Okay, so these particular products are characterized in order to figure out that what might be the proposed or the the actual biosynthetic pathway in order to generate those sort of terpenoids products. Now this is the overall summary. You can see that isopentyl diphosphate or dimethyl allyl diphosphate, these sort of things are either from hemiterpenes or monoterpenes or sesquiterpenes, triterpenes. They are converting to the stepwise process. In every case, those enzymes are operating and they are highly specific. If you get an enzyme which is occurring on that farnesyl pyrophosphate, it will not be occurring to some other kind of products, a substrate basically. So that's why it is one substrate, one catalyst reactions or one enzyme reactions, but products will be multiple. So those enzymes nature are developing in such a way that it will be highly specific to a particular substrate in order to undergo that particular biochemical process to give you a wide variety of the products. So this is the overall summary of this sort of biosynthetic pathway of the terpenes. Now to move on, the sources, you can see that the all sort of fruits, different kind of fruits, that strawberries, they are having different this sort of terpenoids group present, and it can be developed, like they are from the idea that anti-malarial drugs have been developed from there, those terpenoids products. So that is the bridging between those isolations of those natural products, and how therefrom we'll be getting the idea in terms of developing a product or the drug materials to do that particular type of functionals. So it might be the diterpenoids or triterpenoids. You can see, though you should not get scared about to look at the structure, it's too big. But remember that those structures are coming from the nature, and scientists, they have isolated those kind of things, they have purified, they are characterized. Okay, so that's their huge credit without having the highly sophisticated instrumentations. And then only they can classify them, they can understand their biosynthetic pathway, they can develop some new products out of those terpenoid things or those natural products. So that's the like huge significance, like achievement of those particular type of domain, and they are there in different natural products. Isolating them to knowing them and to classify them in a particular domain, you can get the corresponding products out of it. Now to start with the nitrogen containing secondary metabolites. As I said, these terpenes are no nitrogen present. Now once you are incorporating the nitrogen, it is not about the ammonia, you are incorporating the nitrogen atom elements to a particular type of, with the presence of different functionality in a carbon chain. It might be cyclic or acyclic, or aromatic groups will be present. Accordingly, their structures, their different properties, their role, their physical properties, their chemical, physico-chemical properties, those things will be changing. Now to start with is an alkaloid compounds. It's a neutral origin which contains the nitrogen atom definitely. It's a limited distributions and low dosage. It's a marked pharmacological properties of very low dose. Sometimes they might be hugely toxic as well. Okay, so based on that, they are the class of compounds termed as alkaloids. Now to start with very beginning, You can get the acyclic or non-aromatic nitrogen containing 6-membered ring piperidine. It might be pyridine or 5-membered ring pyrrolidine. They are the basic unit of the alkaloid kind of natural products. It can have the tropane kind of derivatives. Now you can see the bridged nitrogen is present in the bridge head. It could be quinoline aromatics. It could be isoquinoline also, the different isomers, indole, imidazoles, or even purine kind of things. Every cases, the common factor is that all are having nitrogens. It might be present in the aliphatic rings, or it is aromatic, or present as a side chain of an aromatic ring. So this way it has been classified, and these sort of alkaloids are mostly basic in nature. Sometimes they are hugely toxic as well. So how it has been developed? What is the historical background? Now the first one, extraction of narcotin and morphine, it was done in 1803. You can remember, you can just feel that how the thing was there. That time you can get it from opium and look at the structures, how complicated that thing is. Now let me tell you something. Once these sort of molecules have been isolated, the characterization itself is a big task on that period. And moreover, whether your characterization is correct or not, that is also another issue, because so many stereo centers are present on that. Now why does things are important there? Because that time there was no work for the chemist. You need to know the nature. You need to understand what nature is doing for us. How come nature is synthesizing a million of molecules on its own way? We are not asking them to synthesize. Nature is synthesizing the molecules on its own way with its own reagent also. You are not providing any reagent to the nature, but there is some particular purpose to synthesize. Okay? So in order to identify them, you should have the proper understanding of their structures, proper understanding of their structure activity relationships, their classifications, their different kind of properties. So that's what there at the very beginning, this sort of natural products isolated from opium, and it is the first to isolate a vegetable alkali. Why alkali? They are having the basic properties. With the nitrogen, they can have basicity, and that is the natural alkali. And afterwards, in 1817 and 1820, two French pharmacists, they have isolated another sort of natural products from caffeine, emmetin, strychnine, quinine, and cinchonine. Now these kind of things are well known. That cinchona, that is cinchona kind of natural products, that is basically quinine, quinidine kind of derivatives. Now there the diversity things are coming. It contains a chiral center. Depending upon that, different class of property will be varied. Okay, based on that, you can see, look at the strychnine structures. They are having total 5 rings. Pentacyclic things are all fused together. So in order to understand their biological activities and their structure, that at that time it was a huge job for the scientist. Another alkaloids is the thing, as I said, that caffeine, nicotine is also been isolated, atropine, cocaine, quinine, morphine. Some kind of cases, some cases they are very good, they are having very defined biological activities, but in other case they might be hugely toxic. And these are some sort of like historical development, how these kind of natural products are isolated with the time and the corresponding the achievements or the recognition of those scientists. Now you can get these are all mostly the plant-based kind of things. But as I said, your natural product can be isolated from some animal source. As you can see, like these kind of things are isolated from snake. The captopril, that is isolated from snakes. You can have that epibatidine, that is isolated from frogs. Now you can understand those scientists, those chemists, they are actually not having any relation. They are not actually the like biologists or zoology kind of things. They are getting the things. They are actually catching frogs in order to get their skin to isolate the natural product from them. Okay, so that was the sort of research on that particular area, followed by the penicillin. I'll come separately about the story of penicillin, how it was discovered, and you all know the huge importance of penicillin for the modern drug discovery. And this way, the animal kingdom, particularly the hormones, different vitamins, they are isolated from different sources. It might be some microbes, it might be some sort of source from the animal as well. Okay, but they have been isolated, they are characterized, they are purified mostly, and their structure was determined that time. So now, this with natural product, you can understand their biological role, but it's hugely limited applications. If you need to get that particular things, you cannot go back to the nature and isolate the product and serve the things, or you can have your own manufacturing unit. That is impossible. So nature gave us the idea where from you have to start, but it cannot go up to the end because you should have the understanding of their structures and which will be implemented further for the drug discovery. So that's why it's a limited distribution in nature. You cannot go back to the nature and isolate them to get a huge amount of those products. That is quite impossible. Followed by the present in the plant, fungi, bacteria, marine, it's all diverse. These sort of natural products which are coming, the sources are of different kinds. That is one, and the physicochemical actions and their poisonous effects. Sometimes you have to develop the technique to isolate them. That is also very tedious. There are different kind of things are involved in it. Biosynthetic pathway of those products are also not properly known. You can have some idea that how that particular, you can go retrospective understanding that if the product is like that, this is the secondary metabolite, how that can be synthesized or how the nature is synthesizing them, you can get the idea, but actual synthesis and things are not being done. So based on that, there are different kinds of alkaloids. These are called true alkaloids, proto-alkaloids, or pseudo-alkaloids. Their structural things are different. Accordingly, we can have the alkaloids which are containing the nitrogen in the heterocycles and originates from the amino acids. So these sort of alkaloids actually coming from the amino acids. That's what they are terming as true alkaloids. Characteristic example, that is atropine, nicotine, and morphine. All are isolated and they are having a similar sort of understanding or the source. They are the amino acids. So that's what it is called the true alkaloids, and as you can see their structures. And the last two, they are the proto-alkaloids. They are not coming from the amino acids, but they can be synthesized or they can be like getting in the biosynthetic pathway on a similar way they can be getting. So proto-alkaloids with the simple amines in the nitrogen atom is not a part of the heterocycles, that might be outside, and they are not coming from the amino acids. The in vivo form of not from the amino acids, simple amines such as serotonin or these kind of things, you see in every cases the amine is outside or it is not a part of or any amino acid naturally occurring thing, it is coming. So they are the proto-alkaloids, or it might be something more, the caffeine, the ephedrine kind of things. You can see it is completely like some carbon chain where the nitrogen is substituted on it. These kind of structural features are there on this kind of like proto-alkaloids. Now coming to the pseudo-alkaloids, most often have been like characteristic true alkaloid, but they are not derived from the amino acids. Though there is having the nitrogen inside the cyclic structures, but they are not similar or they are not coming directly to some, the similar kind of amino acids. That's what they are called pseudo-alkaloids. They are like the structural things are like a true alkaloid, but actually they are not there from the amino acids. These are the structure, these are the different class of alkaloids which are pseudo-alkaloids. Terpenoid alkaloids. We talked about the terpenes. If you have a hybrid structures where the alkaloid part is present and which is related, which are having similar kind of things. Now you can, this second one that probably having the all sort of the cyclic structures with no nitrogen, but only one nitrogen is placed at the outside of the ring. So that's what it's sort of hybrid kind of structures with the alkaloid and terpenoids. That's what it is termed as terpenoid alkaloids. It might be that beta scythenine or the sesquiterpenoids, or like it might be a heterocyclic nitrogen containing substances arising from the metabolism of acetates. So these are basically the representative examples of these terpenoids alkaloids, and as I said, these are mostly used natural products like morphine, reserpine, that colchicine, and that aristocolic acid. They are the structural features. If you look at it, it is the mix of either terpenoids or alkaloids kind of structural features. Now we need to understand their chemical properties. So present in different plant as either salt or ester, because the nitrogen is hugely acidic, it can have their corresponding like hydroxide kind of, or maybe it can have the acidic part of there that will be soluble in water. It can have the N oxide or quaternary compounds, like the nitrogen will be quaternized to have the ammonium salt kind of things, positive salts, and generally they are very much toxic in nature and have the bitter in taste. Their pH values are high, they are basic in nature, and most importantly they are having the light response. They can absorb the light in different wavelengths and can transform to different kind of products after absorbing the light. They are response to the light reactions. Now to have the physicochemical properties which is known from their crystal structures, like it's a solid in crystalline nature. As earlier we said, the terpenoids, they are the volatile, they are fragments, but here they might be very much well crystalline compounds. Exception of the quinine, nicotine, they are liquid, doesn't have the oxygen in their structure in most of the cases. They are only the nitrogenous compound. That nitrogen may present inside the heterocyclic ring, or it may be the outside heterocyclic ring in a long chain carbon chain, and this one. Most of the cases they are the colorless. Exception is there. It is yellow in color, berberine, and the betaine, that is red. They are having very sharp melting point. Why? Because they are crystalline in nature. You can isolate them in pure form, so you can get a very sharp melting point. Most of the cases they can be like a crystal form, be either primary, secondary, or tertiary kind of alkaloid structures. Basicity depends on their availability, because where the nitrogen is placed, because the alkaloids, the basic element present is the nitrogen. So depending upon the positions of the nitrogen, as we have discussed a huge discussions about the pK value yesterday, here also you can get those ideas where the nitrogen group is placed and what is the corresponding conjugate anion after getting that conjugate cation, basically after getting the protonation of the nitrogen. Along with that you can decide what will be the pK value. So you can have those understanding looking at their structures. Their electron donating, of course, electron withdrawing nature can be there depending upon some other functional groups. It might be like type of hybridizations. As I can see that nitrogen, if it present inside the heterocyclic ring, it might be sp2 hybridized, or outside it will be sp3. Accordingly the property of the nitrogen will be changing, and most of the cases they are the heterocyclic ring, they are the aromatic in nature. So these are the features of all sort of alkaloids what has been isolated. Now how to detect the alkaloids? Once you are having the product in your hand, remember that time we didn't have the crystal structures known. We don't have the facilities to go for the NMR, maybe very sophisticated NMR was not there. So you have to devise, but almost it is not that much like earlier where you know the functional group determinations, right? You can do some sort of chemistry on your compound to know the which kind of structures or which kind of like their functional group might be present on it. So accordingly you can see the Wagner's test, you can give their some iodine Ki reagent, you can treat them, and accordingly you can get the corresponding precipitate to get some idea that there might be that kind of functionality present. It might be treated with mercury chloride to have the creamy precipitate of the true alkaloids. It can make a complex with mercury, because those nitrogens are electron donating in nature. They can have the like proper coordinations with the mercury. It can have the Haggard's test with the picric acid. There's a yellow precipitate will be forming from the true alkaloids. It can have the potassium bismuth iodide, the reddish brown property. So these I am talking about. Once you are isolating the product from the nature, you need to know some sort of very basic preliminary test to identify whether the functional groups are present or which way you can classify those natural products into a particular domain. So these sort of discussions we can have a definite test to get this is that particular period of time that you can consider these are the some sort of confirmatory test to understand their structural features. Or you can have the tannic acid solution as well for the different alkaloids to give the colored precipitate. So I can say these are the head book. You are having lot many different products in your hands. You can just taste it and see which kind of colored precipitate are forming. Accordingly, you can identify, you can have some understanding based on their structural features. because there was no other instrument available that time to actually identify their complete structures or their stereochemistry or their other things, even not crystal. So afterward, the process of extraction. So once I am saying the chemist was that period of time, early 1900, they are just identifying some particular type of source of natural products, they are isolating them from that particular sources and tried to like bridge or tried to correlate the structure of those natural products with their corresponding biological activities. So that was the overall objective of their research. So that particular time, the isolation of the natural products is hugely important. And as I said, another things, isolation might not be a very pure compounds. You can isolate the product very easily. Even in the household apparatus also, you could isolate some products from any kind of natural source. But that might not be sure that you can get a pure compound as a single product, because you don't know one particular part of the tree or one particular part of the plants might give you multiple natural products. So that's something very tricky to identify one particular natural product which might be major presence over there on that particular source, and you have to extract it properly, isolate it, and you have to purify it. Without purification, you cannot do the further characterizations. So these are basically the steps of the purifications. Extraction in the fractional extraction, or you can go for the normal distillation process depending upon the boiling point of those natural products. You can go for some other kind of test, like that time different kind of solvent you need to use, because they are polar, non-polar, their polarity varies. You have to test with different solvent in order to understand their solubility. To get rid of the other natural sources like the chlorophylls. Chlorophylls is there in every plant-based leaves and other case. So those leaves, once you are taking the natural products, you have to get rid of the other things what might be present over there, chlorophyll, wax, volatile oils and something. Then you have to do filtration of it, evaporations. Then there are some kind of protocols to treat it with tartaric acid, ethyl acetate, different kind of organic layers that should be isolated, because these natural products what we are talking about, they are all organic, right? There is no metal containing part is present. So mostly they will be soluble in the organic solvents, followed by the aqueous layer that will be, maybe that is aqueous soluble as well. That product is highly polar, that might go to the water level, so that has to be tested as well. They can have the break different kind of salts, like either the ammonia treatment or some bicarbonate treatment to convert them to a salt form, okay? Because salt, if you can make, that will be soluble in water. Followed by aqueous layer, quaternary alkaloids. These are the aqueous layer, or if it is the organic layer, that might be primary, secondary or tertiary alkaloids, depending upon their solubility in water as well as the solubility in the organic solvent. So this is a rough, the steps of the extraction of those alkaloids which are coming from different natural sources, and accordingly you can isolate them. But that is only the first step. You not even purified them. So to move on, maybe I can show you some examples of different structure-based alkaloids. Earlier we say that they are overall property. Now this is the one particular structure, tropane, if you can figure out. This is sort of that structure-based drug discovery kind of things. One tropane, if it is present, then these are the different kind of alkaloids, and all the alkaloids are getting synthesized in a particular type of biosynthetic pathway because their structural features are same. So this particular way you can have an example of huge amount of those alkaloids which is containing the tropane derivatives to get their corresponding synthetic steps. They are the occurrence of different sources. Main alkaloids of this tropane, atropine, hyoscine and that scopolamine, and these are the structures is the major natural alkaloids. Now as I said, their optical rotation value, polarimeter is available but no HPLC. So optical rotation value also says a lot many things, lot many different informations of the structural properties of these alkaloids, and in order to have their some ideas about their structure based on their optical properties. So pharmacological actions and use, if you have the tropane part is present in the alkaloids, they are anticholinergic agents, decrease the salivia and GIT secretions. That time there was no so-called that in vivo, in vitro kind of test, but you can have their idea that they can work against which kind of enzymes. So they can have the antidote to the organophosphorus insecticides. That is also very important because those alkaloids can bind with the phosphorus derivatives, and they are having the mainly for the anti-emetic properties and hypnotic or sedative in nature. Sometimes they are toxic, but with the the functionalizations or to use in different case, you can use them with some particular focused biological activities. So these are the properties of the tropane based alkaloids, and they are the sources. Sorry, you can see like they are basically cocaine derivatives and Atropa belladonna, that solacine, this is the scientific name of that, and from these flowers you can get these tropane kind of alkaloids. Okay, so these are the occurrence of that cocoa leaves that is contain 2% of the total alkaloids. Cocaine, that's a disaster alkaloid, you know the term, it's a narcotics kind of thing, so that is banned as well. But that has been isolated with this particular method, how that thing is there. The main alkaloids of this sort of things are cocaine and the troxylin. These are coming from the cocoa leaves. So this is the structure of the cocaine which is coming from that leaves, and definitely is an abused drugs. Now once it is isolated, that time that was not known to that. So after having their properties, the structures, to know it, that it is having the different sort of which is not good, it's toxic in nature for us, that's what it is, a abused drug it is called. Now quinine kind of alkaloids that is hugely used, that is very good, like use quinine, quinidine, cinchonidine, cinchonine, there are the 4 different kinds of things. They are having huge pharmacological properties, like they are anti-malarial. Okay, they are just coming from the that cinchonine kind of products, like detections under UV. Now another things here important is, depending upon their functionality, their absorbance, the absorption property that might be fixed. You can see that is absorbing UV under 366 nanometer, and the blue fluorescent it is giving with the sulfuric acid. So this is a property that spectral things you can change by treating with some specific chemicals of it. Okay, so that is also an identification things. If you can have the like the absorbance, that is very easy to determine by some spectrophotometer to identify that that's what is the physical property of this compound. So these sort of alkaloids falls under the absorbance of this nanometer, and also they can give a blue fluorescence under this acid treatment. Now some sort of pyridine and piperidine kind of things you can see, like the nicotine, which is also another class of alkaloids which is known as well. So it has been very toxic compounds. These are leaves of the Nicotinia tobacam. That pharmacological effects, it's the nicotinic receptor, highly hydrophobic, so can cross the blood-brain barrier, and accordingly they work. There are different sort of property in the biological systems. Definitely it is toxic, cancer, nitrous amines, and the change in the DNA structure, cardiac disease, it can happen. So the things have been isolated. There is a pyridine based alkaloids, but having different sort of biological activities. Now the question comes, if we know the structure of it, that will be helping. If something is becoming very bad to the biological systems, you can have the idea to devise your drugs without having these structures. So if it is not doing good, that is also very good information to us in order to design the drugs on that particular directions. Okay, so that's what the natural products are helping. So once you are isolating the products, you are purifying them, you are characterizing them, you are well recognized, all the structural, the functional groups are determined. You do lot of different kind of chemical reactions. You can do lot many different kind of other maybe physicochemical tests to understand the structures, their purity, their optical purity, stereochemistry, and if you see that is correlated with the biological activities, that will be helping you to understand the role of those functional groups. That is the role, because you are not actually synthesizing the natural products. Now if you start from the very beginning, this product synthesis you cannot think even that where from why to synthesize this thing. Now once that particular compound in your hand and you can devise, on today's date, you can devise your synthetic technique to these kind of products, and you can design a million of molecules keeping these sort of functionalities intact, right? So that's the bridging part of the natural product isolations and their characterizations in terms of their actual significance in the drug discovery. So this way it goes. We need to know their proper character, like the features. This is also piperidine alkaloid lobeline, biosynthetic origin, that is the botanical sources, and their serine stimulant, that is the cholinergic agonist, that is the premature babies has problem, the respirations, that sort of biological things it can have. So you are just isolating the things and taste, like which kind of things it can give. Accordingly, the high dose cause in a tonic spasm, skeletal and smooth muscle, this sort of property it is having. These are basically very like complex alkaloids. Look, as you can see the structures and having their corresponding biological properties. I think I'm the last example of this. So this is the isoquinoline alkaloids. Now here are some very important things you need to understand. You can see the codeine, morphine, and at the same time that is the heroin. And if you look at the structures, it is nothing but the acetylated morphine. If you do the acetylation of the morphine, you will nothing but will be getting the narcotics, right? The heroin, these sort of things are actually banned. So because of the region, the acetic anhydride, that is even banned. You cannot just go and get acetic anhydride. So if you use it on the morphine, that will be giving you an acetylated morphine, and that is nothing but the heroin. So this particular way, the biological activities are changing in a huge sorry. So overview of the natural products once it is coming from the sources, and what are the steps you have to follow. You take the source material, that might be plant or maybe animal or maybe microbes. You have to extract it. You do the extractions. That will also be depending upon their functionality. So you should have your handbook kind of things, that if the natural product is alkaloid, you have to go for this particular type of extraction. If it is terpenoid or if it is having some different kinds of functionality, you need to tune your extraction process depending upon their functionality. Their solubility will also be varied. Followed by their bioassay, which kind of maybe active compound it is present. Maybe you are having natural products which are not having any kind of biological role. Okay, that is also possible. So you have to test them in different kind of cell lines or some in vitro studies. Then active extract is actually there. Then the important thing is that chromatographic fractionations, the purification stage. So you have to purify then either by normal chromatography or sophisticated instrumentation technique to get a single molecule from there. If it is a mixture of natural products, then your actual purpose will not be solved. You will be getting a diverse nature of their properties, so you will be not be knowing actually it is a hybrid compound or it is a mixture of products or which particular type of functionality it is producing. So the purifications and the chromatographic fractionation, that is very important. Followed by again you have to do the bioassay, because sometimes even we also experience the things. If your natural products is a mixture, then the biological activity is in one way, but if you are taking it in a different class, like different components, and you are again doing the similar kind of biological activities, entirely changing. So that might be a combination effect, because it is some sort of synergistic things. One particular product might work in the biological systems in the presence of another product. So the purification is very important, and after that the active fractions are important. So without the purification and without the isolating of the pure, like the single molecules, the biological activity cannot be correlated properly. So again, after that the isolation, purification, then again one particular compound isolated, and that has to be characterized by different kinds of spectroscopic method, elemental analysis, mass spectroscopy, to know their actual structures. Today's date we are having the single crystal XRD. We can see the actual structure of the product, we can identify their stereochemistry, we can get their elemental analysis, we can also know their optical purity. We are having the HPLC to exactly knowing their enantiomeric excess as well. So these are basically the steps of the overall extraction and overall getting the product from the nature. Because nature is giving a million of products, a billion of products to us. but you need to understand the products in a pure form, to characterize them and to correlate that particular pure compound, single compound, to their corresponding biological activities. So this is the role in between. The chemist play a huge role on that, and afterwards getting the idea from those structures with the correlations of their biological activities, you can design the products and can implement it in a synthetic pathway. So these are the steps. Afterwards, the characterization, since I know the participants, they are very diverse, like they're very vibrant in different background. So I'll talk little characterization techniques of it. So to start with, the natural products, how to obtain that, the elemental analysis. So if you have the natural products, you need to know that whether there is a carbon, hydrogen, nitrogen, you have to do all sort of elements which are present there. So these are the elemental analysis, that oxidations in burning, whether what are the products are coming from there, whether it is carbon dioxide or water or nitrogen dioxide. This way you can do the elemental analysis, followed by you can do the average mass. You can just easily get what is the molecular weight of that product, whatever the product is. That molecular mass, that is very important, but sometimes you can get a fragmented mass. Okay, it is not the actual compound, but a fragment which is coming from that compound. So there might be, you have to tune, you have to like understanding the functional group might be breaking, or there are some kind of bond cleavage will be possible. So accordingly you can get their corresponding structures. So the analysis, average mass, then accordingly how much percentage of carbon, how much percentage of hydrogen, accordingly you can identify that how much big the carbon chain is present over there. Whether the nitrogen group is present or not, accordingly you can identify that whether it's a class of terpenoids or alkaloids, or which kind of property it can give. So this way you can correlate. Furthermore, you can have the mass spectra. As you can see at the end, it is given, you can get a value from the mass. Now if you have the handbook with you, so you can get which kind of secondary metabolites might be close to that particular mass value. Accordingly you can get the carbon chain or the backbone, but functional group, no, you cannot get the actual functional group, whether it is a ketone or carbonyl or an hydroxide. That information you cannot get from this data. You can get the molecular weight, you can get the elemental analysis part, but for that you can go again high resolution mass spectra, which will be giving the molecular weight exactly of that compound, not the fragmented one, but followed by you have to go for the okay, these are the different mass value only, whether these are distinguished between this carbonyl and all. You have to go for the NMR, you have to go for the IR in order to understand the functional group. Then you can have the different kinds of NMR. First NMR will be working on the carbon or hydrogen. You can get how many different carbons are present, how many different hydrogens, what are their positions, what are their shift value. These things are there from the NMR. Followed by you can have the decoupled, that is obviously carbon decoupled NMR you have to do with the proton, carbon decoupled, the proton decoupled carbon NMR you have to do on that. Followed by you have to do the depth, that is the, that know that which kind of carbon it is, whether it's a primary carbon it is attached with like one carbon or three hydrogens, or the even number of hydrogens. This sort of NMR experiment you have to do to understand what is the, the connections of the carbon with different hydrogens and different carbons. So this way you have to stepwise, you have to go. At the end you have to go for the IR, definitely you have to go for the IR to understand the functional groups, whether it's a carbonyl or nitro or the aromatic CH or aliphatic CH, or whether the different kinds of carbon-carbon bond is present, whether it's a double bond or triple bond, or amine or different ethers, esters are present. That functional group identification will be done by the IR, as well as some sort of chemical test. Okay, that already we have talked about. You have to go for some chemical test to identify the which functional group is present. So this is the part about it. Now followed by there the optical property. So once you are getting the compound, immediately you have to take the optical rotation of the compound. Whether it is showing some rotation value, specific rotation value, then that means the compound is carrying a chiral center on that, and that is an asymmetric center. That is a pure chiral center. Okay, so accordingly you can have the corresponding products. Furthermore, you can go for the HPLC analysis for that. So that is also an important parameter to know their stereochemical aspects of your compounds. And then you can have the their UV visible spectrum. This is very conformative things, but that is not a, that is conformative, but that is not a diagnostic things. You cannot get the, you cannot like get the functional group identification just to know the absorbance value. That is not correct. But you can correlate that which kind of the electronic transition is taking place on that, whether it is pi star or n pi star or sigma, which sort of things are happening based on their like lambda max value, the absorbance value, that is very particular for a particular compound. It might not be overlapping, okay? So depending upon that, this physical property also you can validate with the correlations with that particular product. So accordingly, the chromophoric group in UV visible spectrum also you can do. But if your compound doesn't have a chromophore, maybe it's only hydrocarbon, you might not give a very good absorbance on that. So all these techniques, it is not that one particular technique is good enough to identify, but you should have the like these sort of characterizations steps in order to get the idea to either the functional groups, either the carbon-carbon bond, carbon-hydrogen bond on different elemental analysis, or the mass values, as well as their understanding of the functional group. So these are the steps to like the identify or to characterize the natural products. Also, you can have the more sophisticated instruments. You can have the GCMS. GCMS means that compound will be vaporized. It will be going to the gaseous state, and then the mass value will be coming. In order to make it more the mild or these things, you can go for the LCMS, the liquid chromatography mass spectrometry. So that will be the pure compound from there, and in a non-invasive like that is you are not the destroying the compound. That there you can get the mass value. So exactly the molecular weight value could be determined by this LCMS. You can get the LCNMR. You can get the liquid chromatography first, you purify them, and then you can get the NMR. So you will be understanding which particular peak is correlated to which kind of NMR. Accordingly, also you can characterize the things. You can have the different capillary electrophoresis NMR. That is also another purification technique by which it is not a mixture of compound. Okay, so once the NMR, that might give you a mixture of compound. So you will be getting highly purified compound after the solid that extract, and to get a NMR of a single compound. So these are the mostly characterization technique, highly sophisticated, which is available to us in today's date, but earlier we don't have. Once those natural products have been isolated, there is no those sort of instrumentation was available in order to know their structure. Even after more, we can have the single crystal XRD. You can see the molecule actually visualized with us. Okay, so those part are the more sophisticated things. You can see the HPLC, the chiral chromatography. Even inside the HPLC, you can put the chiral column. You can get the peak with respect to a single enantiomer from there. So you'll be knowing whether actually there is any kind of stereochemical impact, or there is no chiral groups are present, chiral center is present. That also could be identified from there. So these are the different instruments, the HPLC, GC, or that supercritical fluid chromatography. That is also a chromatographic technique. In these particular ways, you can first isolate, extract the natural products in some chemical process. You can purify them by different kind of fractionated chromatographic method. You need to identify some kind of parameters like molecular weight, their functional groups, their absorbance, in order to know their structural features. You can get the structures with the NMR. To know the stereochemical like the presence, you have to go for the specific rotations. You can go for the sophisticated like the chromatographic instruments like chiral HPLC or chiral GC, and in this way you can get the overall understanding of their structural features. This is the NMR instrument. You can see like there is a megahertz, there's a magnet present over there, electromagnet, and that is a low temperature. So that electromagnet will work not in the room temperature, that has to be cooled under liquid helium. So that's what that electromagnet will be working in a low temperature, and if you put it, that electromagnet depends like the 400, 500 megahertz, even 700 megahertz, like that magnetic field you can generate over there, but again that will not be possible in room temperature. So that is the low temperature electromagnet will be there, and then you can get the idea about all sort of like the nuclei which present, whether it is carbon or hydrogen or some other, maybe silicon, maybe fluorine, every nuclei could be identified on these sort of things. You need to change the probe of the recording of the NMR, and accordingly you can get the corresponding peaks in this particular instrument. That is a nuclear magnetic resonance. So it's working on the proton, the inside the nuclei, the subatomic particle present. There the interactions of the magnetic moment, magnetic field will be there, and accordingly the signals will be coming. Now coming to the strategies, like so up to now we talked about from the very early, I took you in almost 100 years back in 1800th century, right? Therefrom we have started, and hope you have agreed or you have having like some sort of understanding that how the chemist took a major part in it to isolate and to identify, to understand the different properties, different structures of the natural products, and to correlate them of their further biological activities. Sometimes they are very good to us, they are having different kinds of activities against the diseases, but it may also happen that they are toxic or different narcotics kind of activity they are having. Accordingly, the wide scenario is clear to us. Now, based on those understanding, those structural features correlated with their biological activities, we need to move on to apply this sort of knowledge to the drug discovery. So, to move forward, there are the different kind of plant source if you see. So, extract and then isolate them, exploiting the property of this plant to extract novel medicines inspired by the compound. So, now if you know the structure of the compound which is having the very good biological activities like anti-malarial or antibiotic or anti-cancer property, then you can devise the synthetic techniques, different kind of new which nature may be not giving you, but you can design the things to synthesize those compounds for their targeted biological activities. So this way the bridge of the natural product understanding and for the drug discovery is built actually. So this way you can have different kind of things. I think like we had discussed the secondary screening, preclinical development followed by their further biological activities, and you have to now synthesis once you are understanding the natural products, because once you need the molecules for different biological activities, you cannot always extract from them, but you should have a proper synthetic methods in order to get that compound in your hand. So accordingly you can have this sort of proper steps. So structural elucidation once it is done, but you need to understand the structural elucidation part is most important part, followed by you have to actually synthesize the molecules. That small molecule will be synthesized for further development, then only the natural product will be converted to a drugs. If you are unable to synthesize or if you don't have that, because nature is synthesizing in its biosynthetic procedures, there is no reagents available, but those enzymes actually synthetically you cannot devise. Nature can have the substrate specific enzyme, but we have to do the our work or the synthesis based on whatever the reagents are available in the market. So you have to devise the synthetic tool in such a way that these particular natural products which is good for that particular biological activities should be achieved by doing some particular type of synthetic steps. That has to be there. So up to the characterizations, up to the structure elucidation part, those sort of research was done in early 90s to maybe 2000 also. But nowadays that particular things, that the total synthesis, maybe tomorrow in my another lecture I'll be talking about the present situations of the synthetic, maybe more chemistry I'll be talking about apart from the natural products. We need to understand that you should have a different tools. You know that okay, this is having anti-malarial property, but how to get it? You cannot go to some plants and to ask them that give us the natural products to move forward. That is impossible. You should have every time very smart synthetic tool in order to achieve that particular natural product in a smart way. That is one thing. Secondly, if that particular natural product might have different toxic effects, maybe some side effects, so if you can reduce them, so that's what the drug designing things will be coming into the picture. You mimic the natural products which is giving us the good biological activities, you mimic them, you put install different like the functional group in different positions and try to make a wide variety of the similar kind of molecules, and then you go for the testing. So that's the the natural product based drug discovery. Okay, so accordingly you can see that how many drugs actually 1969 it started from the anti-cancer property, then followed by the antiviral, then again anti-cancer, different kind of things along with the timeline. This sort of drug discovery is happening to understand the structure of the natural products after isolation, purification, characterizations, accordingly to correlate their biological activity, to test their different toxicological properties and all, and then this sort of like the drugs are approved. But again, this drug discovery is not a, you know, the stagnant process. That's a never-ending process, we can say. So today's one drug may be doing good, but there might be lot many side effects. It might affect to some other kind of disease which will be there. It's not a multi-targeted drugs. So we are having, once you are saying this is anti-cancer, but cancer with respect to which kind of cancer? Whether the drug will be administered, whether a cancer patient having the diabetics, is that the thing? So you have to understand the complete scenario of it, because they are very much targeted to a particular type of receptor. They are not multi-targeted drugs, okay? So only concerned, they are biologically active, they are having some focused applications, but they are not multi-targeted. But you can have you can think about the domain starting from there. You can design a different kinds of molecule mimicking that particular molecules. So accordingly the things are changing, and then maybe I can talk about the different kind of products which are actually coming from the natural products and ending to the drug and coming into the market. So this way we have identified the most important product, that is the penicillin, and there are morphine, there are other things as well. This is some kind of data I got, like depending upon their aspirin, their sulfur containing drugs, their FDA approved drugs in 1992. They are the anti-cancer drugs like the Taxol is coming from the natural products. This way the drugs which was earlier natural products, but they are getting a proper destination as a drug. So this way it moves. Now maybe I'll be talking again the penicillin story at the end. These are some sort of like isolation technique. You can see, I don't know, means okay. So that Soxless apparatus, I think this is just for basic understanding of it, just to get the extraction, distillation kind of process. I'll go a little faster on it. Ultrasound assisted extractions, the microwave assisted extraction also you can adopt in order to get the different kinds of natural products which will be coming for that. These are the some kind of like different techniques. Solid phase extraction also you can operate on it, different kind of distillation process. This is something lyophilizing. This is the freezing the things in a very under high vacuum. If the natural products which are coming, that might be degrading during distillation. So you have to do the things under very low temperature distillation. That's the instrument. There is a lyophilizations you have to do. So with that lyophilized, you can get the corresponding product in maybe the solid form in order to get rid of the water or other volatile substance to make it solid, but in a low temperature. So these are some kind of techniques which have been adapted by getting the product in pure form. This is the lyophilizer instrument. You can see the manifold dryer, that the freezing, so pre-treatment formulations. So once you are having the product, you can do the distillations in a very high vacuum under, but in low temperature, not to the boiling point of that particular substance, because it might degrade on that temperature. So I don't think there is some, actually, okay. So here you can see the newly approved drugs which are coming from different sources. It might be a synthetic drugs, natural product derivatives. One is that if you have a natural products, you can derivatize them and get another drug, or you can have directly the botanical drug, what is coming from the nature, or it might be unaltered natural products as well. So you get whatever the structures you are having, that is directly you can use as a drug without having any other further toxicity or some other side effects. So this way, this is somewhat the data for 1981 to 2019. This much percentage of drugs have been approved in the market, or like having the corresponding their activities which is originating from the natural products, or maybe some different kind of botanical sources. These are the small molecules which are coming from there. This might be macromolecules as well. These are the very small molecules of it. Here also you can see around natural products which are coming. Earlier it was little less over here. it is natural product derivatives 18.9%, botanical drugs of that. But now you can see the natural products which are coming from the small molecule part, that is about 27.5%. So these many natural products like driving the drugs are approved in the market up to this 2019. So accordingly, we can have some actual story of the antibacterial agent. These kind of products, though the structures are very big on that, but they are actually showing the very good antibacterial property coming directly from the natural products. These are the things have been isolated, exactly the characterized on that, and accordingly the corresponding activities correlated with their drug properties. So these are the antibacterial drugs have been already there. Natural product mimicking the drugs are coming in the market. Now moving to the antifungal drugs, these are the pro-guard or inactive form of this one. So that's what I'm not going to the detail of their structures, and that I think we have already talked about it. Even not their synthesis, because these sort of things are isolated and their synthesis is also done. Without the synthesis, you cannot go to the industry, because once you will be putting that compound in the market, you cannot ask that go and isolate the product. That is impossible. You have to get a mass manufacturing unit to get that product. Then only you can consider this is as a drug. So these are sort of the antifungal agents which are actually coming from the natural products. So accordingly, the data said the natural product derivatives which are antifungal in nature, about 3% is approved. Synthetic drugs about the 27 in number. Antiviral drugs, because after the that COVID, we can understand the scenario of that. So this way the antiviral drug development also very important in today's scenario. These are the drugs which is coming from different marine sponge. Now here in different kind of natural sources are here, and accordingly the drugs were discovered. These are also the antiviral drugs which is coming. Again, here the synthetic drugs mimicking the natural products is about 21%, which is coming from the different kind of sources, but targeting different virus. How much time we have? Maybe 10-15 more minutes. Okay. So these are the data which are coming from the natural products and converting those molecules at the antiviral drugs. These are the anti-cancer drugs, and in this scenario, Taxol is the actual representative molecules, you can say. So it has been discovered, and then accordingly the different derivatives of Taxol. Now see how big the molecule is. That was from the natural sources, and now you are having different synthetic route of Taxol, and not only targeting this particular drug, it can have a variety like derivative of the Taxol, and accordingly you can get the different kinds of like the cancer you can target with these natural products. So again, the anti-diabetic drugs also we can target from the naturally occurring products. These are the anti-diabetic drugs. You can see sometimes it is the steroidal structures or the carbohydrate-based structures. They're actually coming from the mango leaves. You see this on Mangifera indica leaves, this giving these flavonoids kind of compounds, which is having the anti-diabetic property, which is actually natural products converting to a drug. This is the overall scenario about it, about 8% on anti-diabetic drugs, which is actually been coming from the natural products. Now some microbial or animal sources. Now till now mostly we are talking about the plant sources. Here are some sort of things. It can be coming from the bacteria or other kind of source, and you can see it can come from this sort of like the soil or the prolific producer of antibacterial compound. They are the products which is coming from the source which is animal. Now that's what that epibetidine was discovered in the 1974, isolated from the skin of these sort of frogs, and it is like preparations. So if you go through their isolations, there are many frogs were actually caught or actually captured to get their skins and to isolate the products from the skin of that frogs. So accordingly, their corresponding pain-killing properties is caused by the skin analgesic or pain-killing property it is having this product. Natural products from different kind of fungi. Here the story, the penicillin, I'll be coming in next slides. Accordingly, these kind of things have been developed. Fungi as well. Maybe I'll skip some. This is a marine sponge. You can understand these kind of things are coming basically from the marine algae or marine animals. These have been isolated and they are having huge biological activity which are having mimics to the different drugs from there. Now it can have venom or toxins. Now you can see this sort of snakes, they can have the molecule like captopril are the compound of the anti-hypertensive drugs which have been isolated from the snakes. There is a venom of the snakes that is toxic sometimes, but it is giving the anti-hypertensive drugs from that particular sources. Important natural products for the drug discovery that so this is the morphine which is isolated from the opium, and it's a pain management and opium pharmacology. So this is the story of morphine, which is converted. Morphine is an analgesic which is pain relieving agent is used, but again if you do the acetylated morphine, that is not allowed. So the acetyl like acetic anhydride is banned for that reason. So this is a success story of it, and accordingly the things was isolated, like the early discovery morphine around 1800 centuries, followed by the civil war and then different kind of things widely used. Battlefield injuries, like once this sort of things, those 1800 stage there is no synthetic drugs available. So once your molecule is not giving any side effects, and morphine is a good molecule that is not been toxic also. So been used on that, it's a widely used during the battlefield for the soldiers' disease. That's a success story of this drug. What happened? Okay. These are the different structures: heroin, morphine, codeine, and hydromorphine. So once you know the structures, that has been acetylated through heroin. There is a biosynthetic steps for the acetylations. So you can have the acetic anhydride into it to get the 3-acetyl, and then further it will be converting the synthesis. So you can acetylate the morphine by acetic anhydride to get the heroin. Oh, it's not. It's a Taxol story, discovered in 1960s to 70s, the US National Cancer Research Institutes. So how does the Taxol works? It stabilizes the microtubules. It's working on the different kinds of cancers. That is also a success story. So Taxol have been isolated and converted to the drugs. And not only the Taxol, there are different derivatives, synthetic, semi-synthetic Taxols in the day-wise, like different, like time limit-wise. In 1993, scientists there were semi-synthetic Taxol derivatives, which are also being used. Afterwards, these are the synthetic drugs. So once the Taxol is discovered, the structure identified, then you can go for the Texotrin or the Jevaten. That is in 2010, this kind of drugs were discovered. Now this is the persons who is actually like working on the Taxol discovery. Now we'll come to the penicillins. So you know this penicillin actually is a natural products. It's a blue-green mold. It's a Penicillium notatum. It's coming from there and discovered in 1828 by Alexander Fleming. So that time this molecule is discovered and it is used for the huge antibiotic properties. Okay, so it has been isolated. It has been known structure. Most importantly, this is a beta-lactam ring containing. So it's a beta-lactam is inhibitor as well. So once the beta-lactam ring is present over here, that will be interacting with the protein to cleave that ring, and accordingly the reaction will be taking place. So accordingly the structural features is also very important, and with the time it is like different uses of this molecule was there. So studying the Staphylococcus bacteria, and it's found that that penicillin which is isolated, it is actually killing the bacteria. So penicillin is isolated, which is applied, which is administered on different bacteria, and that bacteria is getting killed. That's the first like identifications of their antibiotic properties. Accordingly, the different type of the things happened. They are having the returning after a vacation that found the blue-green mold, later identified the Penicillium notatum. That's the story of these drugs, and accordingly the bacteria are getting killed. That Fleming was realized to this discovery, and then in the after that 28 September 1928, certainly that did not plan the revolutionize the medicine discovered on the domain of the First World War. So the lot many antibiotic, the bacteria killer was there, and accordingly the things were later in early 30s and 40s. Other scientists was getting involved into it, and then that particular direction was created to the synthesis and to study their drug mechanisms and different properties of the penicillins. %18 of the death caused by the wound related things during the World War I, and accordingly the penicillin was given in 1945. Nobel Prize was awarded to heal that wound healing things during the battlefield, and created a concept in the pharma industry. So it is directly isolated from the natural products, which is accidental discovery. So penicillin was not isolated to treat as an antibacterial effect or something, but somehow it has been seen, it has been found that it is having a property to kill the bacteria. So therefrom it is started, and afterwards the penicillin is coming into the market. There are lot many companies like Merck, that SmithKline, the Novartis, they are all having different kinds of penicillin. So initially you are having isolated the products, you can see their corresponding biological properties, you know their structural features, the complete structure is known. More importantly, their drug mechanism is also known. Because of the presence of this beta-lactam ring, that protein is getting interacted into that, and the corresponding reaction, chain reaction is happening. So if you take out the beta-lactam ring, that is the reason penicillin, it cannot be taken as a tablet. It is always injectable medicines. So that's the thing, is that if you take it as a tablet, it will be hydrolyzed. The beta-lactam ring is hydrolyzed automatically with the HCl is present in the stomach. So it will not be reaching to that particular positions. So it should be injectable medicine. So that is the beauty because of the beta-lactam ring. So that ring will be attacked by the protein which will be coming from the bacteria. So accordingly the reaction is also established. But again, the further modifications are definitely needed. So these are the all sort of penicillin containing drugs, which is synthetic drugs. Of course, penicillin structure is known, and all different modifications in different part of the compound. Now look at the compound is not a very small molecule. It is having bicyclic structures. Most importantly, it is a beta-lactam containing compounds. The 4-membered lactam group is present, and also fused with a 5-membered sulfur containing group. So accordingly the things are changing, and there are lot many side arms. So all the structures I'm not putting here, that's a name. So accordingly you can have the corresponding like the physicochemical properties with their functionality of their structures. So these are some structures, like an ampicillin or that ticarcillin are there. Once there are some kind of troubles, you can just move on to that and then modify the structures to have better biological activities. So accordingly the Nobel Prize was awarded for these 3 persons for the physiology and medicines in the 1945. So that's the biggest story of the natural product derived drug discovery. There is a penicillin as well as Taxol or morphine kind of things. So I think I'll stop here because I have lot many other slides for their synthesis. So I guess maybe I'll talk about on the first part of my next talk, because this is the only discovery part. I'm not talking about any synthesis of those compounds, but after the natural product is isolated, you need to understand the journey is not yet end. Okay, so that natural product cannot going directly to the market unless you have a proper, the tool or technique to develop a huge molecules, huge number of molecules, as well as in a bulk amount. You cannot isolate the natural product to serve the like the necessity of that drug in the market. That is impossible. But natural product will help you out to get the structure, to understand their biology, to correlate their structure activity relationships. Now after that you can design your different synthetic drugs, but there the synthetic chemist will be playing a major role. You should have a technique to reach up to those level. Otherwise you cannot make a proper drug discovery into it. What are the things we discussed like earlier lectures? Structure-based drug discovery, fragment-based drug discovery, they are the biological target. They are having a target which way you can design the drugs. But here you are having the molecules, that the directions are different. Here you have isolated the molecules, just you can understand just a practical. Just like that you are getting a molecule from the nature in free basically. Nature is gifting you. Now afterwards if you find the biological activities, it go back, find out their structures, whether it is a single molecule or multiple molecules, understand their biological role, correlate with their structure activity relationships, then you design your own synthetic methods. You not only to that particular natural products, you can design a different array of the molecules, and then you can move forward for further biological screening. So I think that's it from me in today's session. Now if you have any questions or something, I'll be happy to answer. So please, any questions or something?
I think you have set the base for this.
Yes. Yeah.
Natural products can also act as source of new drugs.
Exactly.
So for that you have set the base.
Yes.
So yesterday and today morning we were listening to the other sources of new drugs.
Drug discovery.
Yeah.
All are mostly synthetic drugs.
Right.
But we cannot forget the origin of it. Like here
Yeah.
from it is started. Once you are having some biologically active compound, that is not that we have started it. We can start definitely, but there will be some basis of that molecule. Where from it is started, and it is very easy for us to get the molecule and taste their biological activity.
Even Professor Stark also talked about the same, that
Exactly.
before we go to in silico designing,
Yes.
we have to work in cerebro.
Exactly.
So this is the same thing.
Yes. Yes.
That we need to have some basis on which we can build on our work.
Design the new molecules. Yes. Yes. So that's the direction of.
It's a nice presentation.
Thank you. Thank you. Yes. And hope you understood that how the steps will be working. That is very important. Otherwise, you won't be having any kind of understanding of the molecules. So the characterization is important here. Extraction is important. Purification is important. There are proper correlations of their structure activity relationship. That is also important. And the thing is that here you are getting the molecule in your hand. Otherwise, you have to design and you have to do all sort of computational studies to do lot that AI-based things, admit analysis. Now you cannot just like that go and get the molecule from the nature. Now today's date you have to apply those things, but we need to understand where from it has actually started. So that's the origin of it. So any questions? Any other questions? Asking? Okay then, that's it. So you can close the session for today.
Okay, thank you, sir, for explaining these concepts in so much detail. Thank you to all of you for joining us today. We will now conclude this session. Thank you.